Similar to the two previous speakers, Michael and Jorge, who talked about various approaches for health monitoring, my group is also very interested in developing technologies for health monitoring. However, the key difference is my colleagues are working on less invasive approaches. So we actually look at bodily fluids like blood, saliva, urine, breath, and we try to look at molecular data from those. And so what I'm going to be talking about today is various technologies that my group and I are developing in this context. So just to give some brief introduction for those that may not have a background in biology, one of the key goals that we have is to develop technologies that are useful for early disease diagnosis through an approach that's referred to as biomarker-based screening. And those that may not have much of a background in biology, a biomarker is essentially any type of molecular indicator that indicates the state of the disease. It signals the presence of the disease, or it indicates whether a patient has a predisposition to getting that type of disease in the future. And so there are multiple different types of biomarkers that we're interested in looking at, molecular biomarkers, that is. One that most of you are probably familiar with are nucleic acid biomarkers, or DNA molecules, RNA molecules, looking at information at the genetic level, which can give you information about whether a patient has a predisposition to certain types of cancers or heart disease down the road, which allows you to take preventative measurements early on. Another type of biomarker which is very useful, which we put a lot of our effort into, are protein biomarkers. So this includes tests that you may be familiar with. So males that have done their physical exam recently, if you look at the blood panel that you're tested for, one of them is the PSA blood test. So that stands for prostate specific antigen. High levels of PSA is indicative of prostate cancer. Also, there's the HCG urine test. So these are like the pregnancy tests that you can buy from Walgreens. You look for high concentrations of the HCG hormone in urine. It indicates pregnancy. And so in addition to proteins, there's also small molecules or metabolites. These are molecules that are less than 800 atomic mass units in size. So there's different challenges associated with detecting these types of molecules. And so in order to make this vision and dream of biomarker-based screening a reality, this requires developing technologies that are rapid and portable, so that they can be used in the clinical setting or by consumers. Instruments that can be worn, or instruments that can be coupled with your smartphones. And so the two key approaches that are useful for doing this is to make the sensing technologies either using electronic approaches, okay, technologies that can be easily integrated with CMOS, easily miniaturized, made lightweight and portable, or if one wants to use optics, move away from the large, bulky instruments, okay, and miniaturize the sizes of the lenses, the light sources, the detectors, so that you can make ultra-compact instruments. And this is something that has been really progressing rapidly over the years. And so, in addition to early disease diagnosis, there's other key advantages to this approach of biomarker-based screening. And one of them is that it enables precision medicine or personalized medicine, right? That is, instead of looking at a patient's outwardly symptoms, there's a much higher probability of success when you prescribe a course of treatment based on knowing the comprehensive molecular condition of the patient. And more importantly, with lightweight, portable, wearable technologies, this opens up the possibility of continuous health monitoring, and the integration of this with what we've been talking about today, as Wade mentioned very earlier, with 5G technologies, is really the holy grail. And so this is really what our vision is. That is to take measurements like my colleagues talked about, you know, the non-invasive physical parameters like respiration rate and heart rate, as Michael talked about, you know, activity, like Jorge talked about, and then combine that information with this molecular data, right? Take things a level deeper, understand the correlation between the two, and in addition to that, understand monitor what we're being exposed to every day, right? Day in and day out, and seeing the correlation between biomarker levels and exposure to various types of toxins. So the result can be extremely powerful. And so really, I think what the holy grail is, you know, the way we do clinical trials, to me, it doesn't seem right. I think we should take inspiration from Yelp. The same way we go to find a good restaurant, we should do crowdsourcing in order to discover biomarkers as well. So imagine we have wearables, you know, millions of individuals, and we're collecting billions of data points. So we have this comprehensive history and this ability to develop predictive algorithms to discover and validate these new types of molecular biomarkers. So I'm going to be talking about three key research thrusts of my group. So one of those involves developing technologies for human health monitoring. Okay, so I'm going to be talking about a technology, a project that we have. It's a DARPA project for doing continuous monitoring of protein biomarkers. And then I'm also going to be talking about technologies for monitoring the effects of environment on our physiology. And then finally, all of these advances in machine learning and data-driven approaches, how can we essentially move away from physics-driven classification and assessment and analysis of biomarker data and move more towards data-driven biosensing? Okay, so these are the three things that I'm going to be talking about. So one of the projects that we've worked on over the years is to develop a technology that can be used to monitor respiratory conditions effectively. So we're collaborating with folks in the medical school where we're developing nanosensors that can monitor inflammatory markers in exhaled breath condensate. So breath has two components to it. There's the vapor phase, right, the gas itself, and then there's the so there's the gas phase, and then there's also the water molecules, the liquid that it's called the exhaled breath condensate. And so if you blow onto a glass or a fog, it's those liquid droplets that accumulate. So that's very rich and full of information. And the traditional way to analyze that fluid, what's going on in your lungs, is what's called a bronchoalveolar lavage. So very invasive. You essentially take a tube that you stick down the patient's throat and take a sample, a biopsy of the inner lining of the airway of the lungs, and you pull it out, and then you use molecular detection techniques. So it's not something you want to do, not something an asthmatic wants to do every 10 minutes, right? Instead, that information is all present in the liquid droplets that we breathe out every day. And so our goal was to use graphene, right, a very novel material that has very unusual properties that offer high sensitivity, but also it's very biofriendly. So essentially take that and analyze nitrite. Nitrite is a marker of inflammation in the lungs. So when you have high nitrite levels in the lungs, it means that your lung is responding. The inflammatory process is essentially kicking off, and something is exacerbating it. So this is something, it's a good marker for asthma. And so we developed this technique. We were able to get very good sensitivity down to the nanomolar levels, okay? And the reason why sensitivity is very important in this case is because as you can imagine, like in the case of a bronchoalveolar lavage, you're essentially sticking a tube and you're taking sample from the lung. But over here, you're breathing it out. So everything gets diluted by several orders of magnitude. So sensitivity is very important. So graphene turned out to be very useful. And we've actually used this sensor to do tests on a series of patients. We did a small preclinical trial. And so there's a need to improvement, but what's very encouraging from our data is for the most part, we're able to rank the order of samples pretty accurately. And so this is one area of health monitoring, which we're very interested in. So another project, which is a DARPA-funded project that we're putting a lot of effort into, is coming up with minimally invasive ways to continuously monitor protein biomarkers. And we're trying to do this in blood. And so you may say, well, blood, that doesn't sound very non-invasive. Isn't there a better option? And the reality of the matter is that blood is, for now, the richest source of reliable health information. Okay, either you have to take tissue, which is very invasive, right? One level above that is blood. I mean, people are looking into things like sweat. We're even looking into saliva. But there's more variation, more fluctuation. And so there's more biological studies at the fundamental levels that needs to really be done before we can advance these devices to sweat and saliva. So blood is a really attractive source for getting reliable information. And so this particular DARPA project, the vision of the whole program is called Electrics. That stands for electrical drug prescriptions. So the idea is, the basic hypothesis is, can you stimulate the peripheral nervous system? Okay, can you stimulate a patient's nerves such that it essentially will trigger the organs to secrete hormones, right, that will treat inflammation. So patients that have rheumatoid arthritis, patients that have inflammatory bowel disease, and other types of inflammatory conditions, can you stimulate the peripheral nervous system to treat? So instead of using pharmaceutical drugs, essentially use a more a safe technique that has potentially less side effects. And so we're not working much on the neural stimulation side. We're focused more on the biomarker monitoring side. That is, developing a platform technology that can be worn, right, that can access blood and monitor protein biomarkers to assess whether the therapy is effective or not. And so essentially this is what a diagram of what our platform looks like. We're collaborating with a group from Penn, Mark Allen, where we are building a flexible substrate that has polymer microneedles on them, and the tips of these polymer microneedles are, we have fabricated on top of them nanosensors. Okay, and if we look in detail to what these sensors look like, essentially we have an array of nanowells where inside each nanowell we have a pair of electrodes that is monitoring the impedance inside the well. So what we have is bioreceptors or biorecognition elements that are very specific to capturing the protein that we're targeting. So as blood is running over this sensor and we're capturing the target protein, we're measuring increases in impedance between these electrodes. And so the advantage of this is that it's a label-free technique. So traditional methods for analyzing proteins, you essentially have to use fluorescent dyes and use big bulky optical scanners in order to look at a single time point at a time. What we're proposing here is something that can be worn that can do continuous monitoring. Take one time point every 10 minutes. And so we have fabricated these devices extensively. We've characterized, the different geometries that improve sensitivity. So the smaller the well size, the higher the sensitivity. As you can see over here, 2 micron size wells gives a larger drop or larger change in impedance when current binds compared to 4-micron wells. We've extensively benchmarked our technology with respect to the gold standard. So Luminex and ELISA are gold standard techniques for analyzing proteins. So we have comparable detection limits and comparable dynamic range over 4 to 5 orders of magnitude concentration. And the advantage here is, instead of doing a 2 to 3 hour long test with a benchtop instrument, we have a miniaturized portable test, which is one step and requires only 6 minutes. Okay, so this is something that can really be potentially enabling. And so we're collaborating with Hubert Lim at University of Minnesota and Medtronic, where they are doing ultrasonic stimulation of the spleen of patients that have rheumatoid arthritis. And we are using our sensors to test biomarker levels in the blood. And so what's very interesting is that we're very easily able to use our sensors to distinguish between healthy patients and arthritic patients. So these are actually animal studies, but we're also doing human subjects as well. So our data for animals look like this. And also, when we look over a cohort of 20 animals, you can see that the amount of swelling in the mouse's ankle, so if you look at the diameter of the ankle, you see increases in the biomarker levels as well. So we look at different biomarker levels, cytokines, so IL-6, TNF-alpha, IL-10, so these are markers of inflammation in the blood. We're also we're doing both in vitro tests, where we take a prick of blood from the animal and we place a drop on our sensor. We're also building in vivo sensors where we have microneedles that we are piercing into the tail vein and also the thigh muscle of the mouse. I know you're looking at this and saying, well, this doesn't sound very non-invasive, but you have to start from somewhere. I know this doesn't look very appealing as a wearable device. Probably doesn't make any of you want to wear one of these, but just wanted to show you we're really doing experiments in animals. And so, but what's promising is that the results that we see in animals correlate very well with the in vivo experiments correlate very well with the in vitro experiments. So in both cases, here we have a mouse that naturally expresses high levels of this inflammatory molecule, IL-8. We get rises in impedance when we inject these sensors into the mouse's tail vein or the thigh muscle. But in the case of a healthy mouse that has low levels or non-existent levels of IL-8, you essentially see a drop in the impedance. So we have also worked to miniaturize not just the sensing apparatus, the sensor itself, but the whole footprint of the platform, the lock-in amplification platform, and coupled it with Bluetooth and developed a smartphone app that essentially performs the signal processing and display of the data for the user. So here we fabricated this lock-in amplifier circuitry onto a flexible printed circuit board that can be worn by the subject. And so here we couple this over here with cytometry data. Another aspect of the work, how much time do I have, by the way? I know we started late. I just want to make sure I don't go over time. About 5 to 10. Okay, thanks. So another aspect of our work is that we're collaborating with the Cancer Institute of New Jersey, Joe Bertino. He's an oncologist, where his group is developing a novel type of therapeutic that instead of just giving a patient chemotherapy drugs, which not only kills the tumor, but it also kills healthy cells as well, they're using they've developed a much more targeted approach. So essentially what they do is they're coupling these chemotherapeutic agents to antibodies. So antibodies are molecules, proteins that are very specific. They bind only to the tumor cell. And so when you do that, what's happening is you're delivering the chemotherapeutic agents just to the site of the tumor, as opposed to killing other cells, which results in so many different types of side effects. And so our objective is to try to come up with a tool that can assess whether a patient will be responsive to this type of therapy or not. Okay? And so this is challenging because this antibody, it binds to a particular surface protein that's referred to as matriptase. But not all patients, not all cancer patients have tumors that are expressing this protein. So it's only effective for those patients that have tumor cells that express matriptase. And so what we're working on is developing a sensing platform, microfluidic sensing platform, that can rapidly assess, take a sample of a patient's a tumor biopsy and assess whether the patient's tumor cells do express matriptase or not. Okay, and so by doing this with a portable, rapid electronic analyzer, now you can imagine doing this type of assay either in a doctor's office or in the operation room as the biopsy is being collected. So the approach that we're taking is to take the biopsy from the tumor biopsy from the patient, and then essentially take these therapeutic agents and incubate them together with the cells to see whether the cells are still alive or dead, right? And if the cells die, then that means that the patient's tumor is also expressing the matriptase. And so the state-of-the-art technologies in order to do this are essentially cell viability analyzer. So this is a large, bulky instrument, costs tens of thousands of dollars, not something that every mom and pop doctor's office can afford, right? So the question is, how can we miniaturize this and make it into something that's portable and rapid, but accurate at the same time? And so this is where we thought it might make sense to utilize impedance cytometry. So the way that impedance cytometry works is you have a small microchannel that has a pair of electrodes that you flow cells through. Okay, and so as each cell passes over the electrodes one by one, you get a peak. All right. So normally, in this community, the approach that researchers use in order to try to classify cells is more of a physics-based approach. All right. Well, we thought this could be a very interesting data-driven problem, because you can do these measurements at multiple frequencies. Okay, so the dielectric properties of cells, they're frequency dependent. Okay, they're very hard to characterize physically. Right? Cells have so many different proteins and DNA inside them, different types of molecules, different conductivities. It's very hard to physically characterize and model accurately. And so it makes sense to use a data-driven approach, because you can easily get measurements of thousands, even tens of thousands of cells within minutes. Okay, and so we fabricated these devices. Here's an example of the microscopic view of what our cytometers look like as a cell passes by the electrode pair. And this is an example of a schematic of the circuit. We use a lock-in amplifier circuit. Michael already talked about obtaining I and Q. Essentially, we have an amplitude demodulator circuit over here that locks into particular frequencies. So we do this over 8 frequencies. So we get a snapshot of the cell's dielectric properties at 8 different frequencies. And so here you can see, as one cell passes by, an example of the peak at 3 different frequencies, right? So you can see the size of the peak differs depending on what frequency you're exciting it with. And so if you look at this data at multiple frequencies, you can see that there is this ability to start to differentiate between the two populations of drug responsive cells, drug sensitive cells, and drug resistant cells. Okay, so here is data of amplitude change at 500 kilohertz and 20 megahertz, and this is at 30 megahertz versus 500 kilohertz. And so here we look at the whole spectrum to try to better understand which frequencies are good features to use for our machine learning classifier. Okay, and so you can see that there are certain frequencies where it's more easy to differentiate between the two cell populations compared to others where there's a lot of overlap. And then we've done benchmarking with respect to the gold standard. We've done this on a small subset of samples. We're still working and doing more, but the initial results look promising. Overall, we're able to do a decent job of predicting. In this case over here, you can see the results are off, but hopefully with more and more testing, we'll be able to essentially better understand how good the accuracy of this is. So I'd like to just end quickly, since this is an industrial advisory board. I thought it would be interesting to sort of kick off this conversation. What are the barriers and impediments to getting this type of technology out so that consumers and patients can actually use them? And one argument that I like to make is that biosensing technologies and lab-on-a-chip technologies like the ones that I've discussed actually have a difficult time. And one of the reasons is because there's no such thing as a free lunch, right? As you shrink down the size of the sensor, the amount of fluid that you're analyzing, you tend to lose accuracy and precision. It's the Elizabeth Holmes problem, right? You can't you can't get everything in the world with a drop of blood. So that's one challenge. And so after 30 years, ELISA, the gold standard, the big bulky instruments, that's still the gold standard. You go do a blood test at the clinic, they're going to do an ELISA, right? And so the question is, why not, right? All these great technologies that people are publishing in the lab, why are they not making it to industry? And one argument that I like to make is that, if I get a $300,000 or $500,000 career award from the NSF, we're happy and we celebrate, and that is enough to get us to a point where we show proof of concept. But to really get this to the point where you can start to sell it, you have to go through FDA approval, right? You have to go through this process also of CLIA waiver. So you have to essentially, this can require $5 million, $10 million, even $20 million in capital to go through this process, right? So FDA approval, you have to go through several hundreds of patients showing that your device is as accurate as the gold standard, a predicate device. CLIA waiver is essentially showing that your device is idiot-proof, so that any person on the street, as long as they read the instructions, they'll be able to test it and get correct results. So this is challenging, and so this tells me that this connection with industry needs to happen very early on in the game, as opposed to us doing our own thing in the lab, and then after our grant ends, not knowing what to do with it. So yeah, I think I'm probably out of time now, so I'll end there and be happy to take questions.